This is Lauren, Alex, and Zikora of Pod B from Team 3 in Putting Sleeping Sickness to Sleep, or an investigation into the elimination of African trypanosomiasis through the alteration of the vector Glossina morsitans via addition of the human OGT gene and utilization of CRISPR gene drive.
In this project, we are attempting to remove, or at least mitigate, African trypanosomiasis, also known as sleeping sickness. This is a potentially fatal disease caused by the trypanosoma parasite that uses the native tsetse fly as a vector. Although the infection is not currently found in the United States, it is a serious problem in 36 sub-Saharan African countries. Effects are felt throughout the continent, but the worst of the disease is seen in Central Africa. As you can see in Figure 1, the only countries that are not affected are those at the very northern or very southern tips of the continent. In terms of the disease, victims can expect mild to severe consequences, depending on if they receive treatment or not. In the weeks following infection, the central nervous system is attacked by the parasite, causing mental deterioration. If left untreated, the disease results in coma or death. There are more than 10,000 new cases reported each year, and more than 80% of deaths that occur happen within the first 6 months of the illness. Current drugs used for treatment are considered highly toxic and be difficult to administer. Because of these difficulties in administering drugs, a genetic approach in the fly vector will be beneficial to reducing the amount of people affected by this disease. A genetic approach would also benefit people who are unable to receive treatment, whether that be due to location or poverty. Africa is the poorest country in the world, and they suffer heavily from this disease due to a lack of treatment availability. Figure 2 illustrates how some of the poorest countries in Africa have been found to have the highest densities of this fly, and therefore this disease. However, it is not just humans that are affected. Livestock is susceptible to this disease, and this causes mass death and loss in valuable product and economy value for these poor people. There are devastating impacts on the livestock and African economy. The statistics shown on the slide show just how bad it really is. The tsetse fly was selected as the target for CRISPR-Cas9 daisy chain gene drive because it is one of the most prevalent vectors of African trypanosomiasis. The genome of this species has also been sequenced, making genome editing simpler. For the genetic modification, the OGT gene found in humans would be inserted into the tsetse fly. This would be inserted into the fly's genome along with a CRISPR package which encodes for a gene drive. This CRISPR package includes the OGT gene along with a predetermined number of sequences which encode for more CRISPR packages. This allows for the modified organism to have a nearly 100% chance of passing down the modified trait when mating with a wild-type individual. One CRISPR sequence is lost with each generation, meaning that in order to control the range of the genetic drive, eventually it ceases to spread throughout the population at a non-Mendelian rate. The OGT gene encodes a glycosyltransferase which catalyzes an N-acetylglucosamine to serine or threonine residues through O-glycosidic linkages. A study conducted by the University of Bristol and the University of Aberdeen found that infection of tsetse flies by the trypanosoma parasite is greatly reduced with lower concentrations of N-acetylglucosamine. The purpose for the insertion of this gene would be to change the structure and function of the N-acetylglucosamine already in the tsetse fly cells, as it is the second most abundant carbohydrate. This would lower the concentration in the tsetse flies and reduce the rate at which tsetse flies are able to become vectors for African trypanosomiasis. Most replications of the trypanosomes occur in the tsetse fly midgut, and the human OGT gene has a high expression rate in the gut, so it could hopefully stop the infection at the source. Another study concluded that carbohydrates in the midgut inhibit the trypanocidal effects of lectin secreted in the midgut of the fly, so that is why one could expect that the reduction of the N-acetylglucosamine would create lower infection rates. There has not been any gene drive previously performed on tsetse flies with the intention of eliminating African trypanosomiasis, so the project is quite novel.
So for our methods, our first step will be to do a PCR, which is run to isolate the OGT gene from the human chromosome 10, and we'll use the forward and reverse primers shown. The BAM-H1 and BSM-BI restriction enzymes will cut the DNA at specific sites. We will also be using the gene drive method to continue the expression of the OGT gene in the future generations of flies. Gene drives are DNA sequences, and they act as self-propagating mechanisms of spreading a genetic variant through a population. They work by inserting themselves in homologous chromosomes, which will lead the offspring to also inheriting the drive. This is achieved by the insertion of a sequence which encodes the creation of more richer packages. Next, we will put the gRNA oligoscaffold into the p-lenti cascade vector to encode the endonuclease enzyme Cas9. The gRNA scaffold codes for a guide RNA which will locate the precise section of the fly genome that we want to insert our OGT gene into. It will direct the Cas9 there for editing, and the Cas9 sequence will encode for the Cas9 enzyme which will cut the fly genome. The U6 promoter to express gRNA, the CMV promoter to express Cas9. To set for the gRNA sequence within the plasmid, insert the gene drive sequence, which encodes the replicable CRISPR-Cas9 system. This will allow for the gene drive to take place, and we expect a near 100% inheritance rate of the OGT gene and the gene drive system to be achieved. The forward and reverse oligos are shown on the slide, and there are overhang present on two strands to aid in the cloning process. The overhang on the forward oligo is CACC, and the reverse overhang is AAAC.
First, we want to transform the vector into competent E. coli that we obtain from the Carolina Biological Supply Company. We're going to genetically alter a cell by the process of heat shock. First, we'll create a sudden increase in temperature, which will create pores in the competent E. coli. These spores will allow plasmid DNA to enter the bacterial cell. Further, we can identify which cells have transformed by placing the cells on a chloramphenicol plate. The cells that contain the CMR gene present in the vector will survive. Next, we will micro-inject the plasmid from the E. coli into 3,000 testi-fly embryos obtained from the Seattle Children's Parsons Research Lab. We believe this will work because a process like this has been performed before. However, in this case, they micro-injected the vector into sandfly embryos. This provides proof that our experiment is feasible. Now, we can see if our experiment was successful by performing diagnosis of the testiflies. We'll diagnose these testiflies for the presence of the protozoa. We'll extract DNA in order to isolate the protozoa, then perform PCR to amplify this DNA, and finally gel electrophoresis in order to determine the presence and prevalence of protozoa in the given organism. For this part of the experiment, it is important to have two groups. The first group is the flies that have the OGT gene, and the second group will be the flies without the OGT gene present. This process is important in seeing the success of the OGT in the testiflies. In the gel electrophoresis presented here, it can be used to determine the testiflies are hosts for the protozoa, and also we can identify what type of protozoa is present.
The AMPR promoter and CMR selection marker will be used to make sure that the plasmid is inserted into the transformants. The AMPR promoter will ensure that the CMR selection marker gets placed into the plasmid. The vector will be transformed into the E. coli using heat shock and then plated on plates containing the markers. The cells containing the CMR gene will survive, meaning they are transformed, and the untransformed cells will die. These transformed cells will be then inserted into the ZT flies. The GNR scaffold sequence is necessary for the Cas9 binding. The gRNA scaffold codes for a guide RNA which will locate the precise section of the fly genome that we want to insert our OGT gene into. The BAMHI and BSM-BI are restriction enzyme sites where the DNA is cut, and the U6 promoter will allow the OGT gene to have high expression of the fly, and the CMV promoter is used to get strong expression of Cas9. The CRISPR encoding gene drive package is a sequence which encodes the CRISPR system. This can be created by the cell, allowing for the editing of homologous chromosomes and demonstrate a non-Mendelian inheritance rate. The location of the package does not matter as long as it is behind the U6 promoter, allowing it to be highly expressed in the fly.
Now looking at a phylogenetic tree that traces the evolutionary history of the OGT gene, We can tell that this is a gene expressed in Homo sapiens, which are humans, and not in Glossina morsitans, which are the tsetse flies. This could be a problem, because these two organisms are not closely related. And since we plan to introduce the OGT gene into the fly, this might not be successful for a couple of reasons. For one thing, the gene might not be able to be expressed, or might be expressed in an unexpected way, such as negatively impacting the phenome of the fly. Furthermore, if we are successful, we know that the OGT gene is constantly transcribed, and this could be bad for the flies, because there is an energy expense of this constant transcription. This constant transcription could lead the flies to be selectively chosen against, and the GMO flies might die off quickly, producing very little generations. In our experiment, there are a few ethical concerns. There is a possibility of the transformed tsetse flies being released into the ecosystem, and this could have unforeseen consequences on the ecosystem. In order to control the release of the flies, we will design proper containment. The containment vessel will include multi-layered netting and container, and there will be restricted access to the container. Furthermore, there is a concern of adding human genome material to the insect genome. However, GMOs are relatively common, and if we follow the proper guidelines when dealing with the flies, we can negate any potential risk. There is a possibility of the faulty gene in tsetse flies interfering with current fly population. However, we don't plan to release the flies for now. We just want to see the effect of the OGT gene on the protozoa in tsetse flies. Finally, there is a possibility of the OGT gene entering the gene pool, but by successfully containing the flies, we can stop that. In this experiment, we'll be careful in ensuring proper containment of the flies, because if the flies gets into the environment, it could be terrible. As of now, we don't know the effect that these flies might have on humans, so proper containment will be important.
For future directions, a possible future direction includes the testing of livestock, including sheep and cows, with the GMO tsetse flies and determining the rate of infection via a control of non-GMO flies. This process will allow statistical measures, obtaining and caring for livestock, as well as oversight on the ethical treatment of said livestock. This experiment would illuminate how the GMO tsetse flies can actually decrease the rate of infection due to the decreased number of parasites as a result of the OGT gene. Another possible future direction could be the application of the CRISPR daisy chain drive. This daisy chain drive can be utilized to limit the spread of the GMO gene should it be introduced into the environment. This is a result of each generation losing an aspect of gene drive from the end of the non-driving chain. This results in a decreased expression of the CRISPR package until the gene is no longer expressed. This process would allow for proper containment as well as guards against the added OGT gene mutating as it's passed down, which could result in more negative impacts on the local ecosystem. Here are our references.